Yeah, so as far as this week goes, the lab this week is a review lab. So you will review everything to get ready for the practical. Okay. Part of that will be having a chance in one way or another to look back at some of your quizzes, the lab quizzes from the 3 anatomy lab quizzes we've had. Part of it will be actually getting the fetal pigs out and the sheep hearts out and looking over those. Okay, so bring your dissection kits. Some of you, if your dissection isn't perfectly nice, which I'm sure not all of them are perfect, or most of them are not perfect, then you'll have a little bit of time to kind of clean that up, because we do use the specimens that you dissected on the practicals, right? And so if yours is nice and clean, then there's a good chance that yours might show up on the practical. So there'll be a little bit of time to do that. Also make sure to bring your textbooks to lab this week, okay? Because there'll be some review. You can review some of those figures and diagrams in the textbook, as well as some of those the images, or not the images, but the heart anatomy. Why can't I think of the name, the word for those? But the heart models, the heart, yeah, those models in there. So we'll just get a chance to review all of that stuff. I will actually be in Professor Mahoney's labs, and so the morning and afternoon labs I'll be in there. Yeah, and then you'll still have Dr. Silva for the night lab. Okay, I think I saw a question on the chat there. Yeah, so Rachel's good question there. We'll be using pictures of the cadaver. Oh yeah, definitely not. You won't be using the cadavers. It'll just be images, and most of those images for the human will be similar to the ones in the textbook. It looks like the drawing type ones, not the actual like pictures of a cadaver. It'll be the drawing or like diagram type ones. Okay. All right, and we'll walk through a little bit more details of the practical in lab on Thursday too, as far as the flow of the stations throughout and all of that. Okay. And then so what we finish today in class will be the last material on exam 4. Well, and then Wednesday's class will move into the section 5, and then we'll have exam 4 on Friday. Okay, so next week's lab will just be that Tuesday meeting, whatever that time is that you signed up for, and you won't have a regular lab on Thursday next week. Actually, this lab quiz this week is your last lab quiz, because of the way it works out. So this will be the final lab quiz, and it'll be over the stuff we did last time with the blood lab. So yeah, so it'll be over. Just make sure you understand what all the different cell types are in the blood and what they do. Make sure you understand the different causes of anemia and kind of what that would look like, and just kind of make sure you understand how to do those different measurements, like the hematocrit measurement and some of those things that you did with the blood, with your own blood in the lab. And you should be fine for the quiz. Okay. All right. Any other questions about anything going on this week? I have one more question here. Yes, lab practical is in the same room that you have lab in Seder 117 there, Juliana. Okay. All right. Great. All right. Well, let's hop over to the notes here. And so today we're talking about carbon dioxide and how we control ventilation. So we're going to start out with a question here. Let me see if this opens back up. Thing might just be giving me major trouble. Says it's open, but I can't find it. Sorry, let me try one thing. Actually, I'll get this passing around while I'm trying to figure that out. Okay, see if that did it. There it is. Okay, Gosh, so I think maybe we can actually see your word cloud now. There it is. No, okay, we won't do that. All right, don't push my luck. It's gonna really mess up on me. Okay, let's try this question then. All right, so yeah, go ahead, answer that question. Our major system used to buffer excess hydrogen ions works by us releasing which chemical into the lungs? All right, just give you about 10 more seconds. Just go ahead and take a guess if you're unsure there. Oh, and by the way, you should have received all the emails with your feedback for the peer evaluation. I just sent those out right before class. So yeah, so just take that as feedback for the next TBL thing that we do. All right. Okay, let's see what you guys said here. Okay, so about 70% of you are saying CO2, okay, which is absolutely right. It's definitely CO2. The kind of strange part about this is there's no hydrogen in CO2, right? So we're getting rid of hydrogen in a way by releasing that CO2, okay? And that's all dependent on the main chemical buffering equation in our body, which you'll see here in just a sec. Okay, so CO2 transport. Much more of it is dissolved in the plasma than we do with oxygen, so about 10% is carried that way, okay? It can also bind to hemoglobin, okay? It binds in a slightly different place than where the oxygen binds, but it can also be carried by hemoglobin. That's only about 20%, so that takes us up to about 30% of our CO2 being carried by those two methods, okay? And then one thing that we talked about before is what happens to O2 binding when CO2 is bound to hemoglobin. Okay, this is part of shifting that curve that we talked about, right? The higher CO2 levels lead to oxygen coming off of hemoglobin more readily or increases unloading. So that's what we're talking about, right? So the O2 affinity of hemoglobin is reduced when CO2 is bound to hemoglobin. Okay, so the more CO2 there is present, then the more likely oxygen is to get pushed off of hemoglobin. Okay, but then the majority way that the CO2 is carried in the body is by this bicarbonate ion, and that is HCO3 negative. Okay, so these are the three primary ways that we carry CO2 around the body. It's a little bit more complicated of a story than it was with oxygen, right? Oxygen is 98% is carried by hemoglobin in the red blood cell. CO2 is a little bit more of a mix and match different ways to do it, but still the majority of it is carried as a bicarbonate ion, okay? And so this is all centered around this idea of this buffering system as well. And we looked at this buffering system last semester. So the most important buffering system in the body is our bicarbonate buffering system. This is definitely the primary buffering system of the extracellular space in our bodies. Okay, and the equation for that is right here. We've seen this equation before. So we take that hydrogen ion with the bicarbonate ion on one side. We have carbonic acid in the middle, and then we have CO2 and water on the other side. Okay, and this can happen. This reaction happens in two primary locations. It can happen just right out in the plasma in the blood. Okay, it doesn't happen very quickly when we do it that way because we don't have this special enzyme that speeds it up called carbonic anhydrase out there in the plasma. But then we can do this same reaction in the red blood cell at a much faster rate because that's where we have that enzyme carbonic anhydrase. Okay, and when we're talking about the kind of the buffering function of this equation, this is kind of back to the very first class of Bio 1030, right, where we talked about what happens if we put a strong acid into the solution that has this, does it shift to the right or to the left, right? So when pH goes down, right, meaning that we increase the number of hydrogen ions, which way does that equation have to go? It has to go to the right. Okay, because by going to the right, that means we're taking hydrogen ions and binding them to bicarbonate. So we're kind of holding on to them so they don't affect pH anymore. Okay, if pH goes up, meaning that we don't have enough hydrogen ions, then this equation needs to shift to the left so we can add more hydrogen ions to the solution. Okay, but that's kind of a separate thing. We're not talking so much about the buffering side of it now. We're going to talk more about the CO2 transport side of it now. Okay, any questions on any of this so far? Okay, so really what we'll talk a lot about today is this thing called the chloride shift. Okay, and the chloride shift is something that happens in the red blood cell that's very important for transporting CO2. Okay, and what this does, it allows this bicarb reaction to happen in red blood cells. And there's an exchange that happens. Okay, so there's always going to be an exchange between chloride, Cl negative, and the bicarbonate ion across the red blood cell membrane. Okay, when one goes in, the other one comes out, and when one goes out, the other one comes in. Okay, so why do we have to do this exchange? Because for one, the primary reason is if we were just releasing a bunch of negatively charged ion like that bicarbonate ion outside of the cell, then we would disrupt the membrane potential of that cell, and that would change some proteins, that would mess with some things in that cell. So if we release the bicarbonate ion, then we bring in an also negatively charged chloride ion, and that maintains the membrane potential. Okay, so the direction that this happens, if this red blood cell is down at the tissues, okay, then this is going to be that the bicarb goes out of the red blood cell, chloride goes in, because this is going to help us deal with high CO2 concentrations that we have down at the tissues, because that's the tissues are producing that CO2 as a byproduct. Okay, and then when that red blood cell makes its way back up to the lungs to be released, to release the CO2, then we do the opposite. The bicarb comes into the red blood cell, chloride goes out, and this allows us to release CO2 in the lungs. Don't worry if you don't have that all worked out in your head yet. We're going to spend a little time with it over the next few slides. Okay. All right, looks like everybody's done writing. Okay, so here's what I want you to do. Excuse me. The next two slides show a red blood cell, okay? And then they have some questions there. Okay, so where in the body is this? I just told you kind of either the tissues or the lungs with that we see a certain direction of that chloride shift. Okay, so I'd like you to work with the person next to you, if you're here in class, or the people in your breakout rooms here in a second when I send you there, to answer all of those questions on these next two slides. Okay, so where in the body is this? That's just tissues or lungs. Okay, where would we see carbonic anhydrase working? What happens to the hydrogen? There's a lot of questions here. Okay, so just kind of a lot of questions for discussion. One thing you can also do is I mentioned 3 different ways that CO2 is carried in the body. All 3 of those are represented here in these images. Okay, so you can kind of identify those. All right, and yeah, so just spend some time discussing these things on the next 2 slides, and then we'll come back together in a couple minutes. Thank you. Andrea, did you get this? Yeah. Okay. Oh, I'm just looking at one back. Okay, yeah. Well, you signed the one back. Maybe it did. Yeah. Okay. That's it. That's fine. I got it. Yeah. It doesn't matter. It's okay. I got you. Good. You good? Okay, so there's a lot on these two slides. Obviously, a lot of questions, so you might not have gotten through all of it. That's okay. Let's go ahead and talk through some of this. So we've got these two different ones here. We'll start with this first one here. Okay. So where in the body is this? If we got the options of the tissues or the lungs, why don't you guys on Zoom go ahead and put that into the chat? You just type in either tissues or just do a T or an L, tissue or lungs. Good. Pretty much everybody's saying T for tissue. You guys in the classroom here agree with that? Okay. Yes. Okay. So this would be the tissues. Okay. So we see the CO2 is being produced by these cells, right? And it's being sent out, right? That's a byproduct of doing metabolism, right? We talked about that metabolism, the energy systems last semester. CO2 comes from the Krebs cycle, comes from the production of from going from pyruvate to acetyl-CoA. Okay, it's a byproduct, right? So we're got CO2 coming out. Okay, so that's one way to tell. We can also tell by the direction of the exchange too, right? In this one, chloride is going into the cell or out of the cell? Which one? Going in, right? Yeah, we can see it going in. Okay, and that's just what I told you here, right? So at the tissues, chloride goes in, right? Okay, so you could have figured that out that way too. Okay. All right. So carbonic anhydrase, that works about right here. Okay. We'll just say Ca there for carbonic anhydrase. That's helping to drive that reaction. Okay. Inside of the red blood cell, but it doesn't happen out here. So we don't have carbonic anhydrase working outside. We have that same reaction happening, but it's just a lot slower when we're doing it out there. Okay. So we'll just say in red blood cell. Okay. So what we see there, what happens to the bicarbonate ion? Okay, because if we do the reaction this way, we end up creating, not the bicarbonate ion, what happens to the hydrogen ion? We end up creating a hydrogen ion, right? And we wouldn't want to mess with the pH too much by just dumping a bunch of hydrogen ions out into the blood. So what do we see? We see that it binds the hemoglobin, actually, right? Hemoglobin is a protein, right? And we talked about buffering last semester. And you guys have talked about it in chemistry, that one of the buffers in the body is just proteins, right? Hydrogen ions can bind to proteins. And so hemoglobin is an example of one that can act as a buffer. Okay, so we make this hydrogen ion, which could potentially be a problem if we just made a bunch of it and just dumped it out into the blood, but that's not what happens. It gets bound to the hemoglobin instead, right? So we'll just say that it gets buffered by hemoglobin. Okay, and then what happens to the bicarbonate? Well, the bicarbonate is sent out of the cell, so released out of the red blood cell, okay, and it goes out here into the plasma, okay? And so we're already in a situation here down by the tissues where we may have the pH kind of dropping a little bit anyways. And so sending a little bit more bicarbonate out into the blood isn't going to be a bad thing, right? Because this is actually just helping us maintain pH, okay? So we got that. And then we see, where are those three methods for transporting CO2? We see in the form of bicarbonate both here and here, okay? But that happens both inside the cell and out of the cell, okay? So that was our 70% one, right? So we'll just say 70 there. Okay, then there was one that was just dissolving into the plasma. That was 10%, right? That's this one right there. So just CO2 going to CO2 in the plasma. Okay, and then we see CO2 binding to hemoglobin. That one was the other 20%. That's right there. Okay, so we've got those three methods of CO2 transport. Okay, and what happens to chloride? Well, chloride comes into the cell, all right, into red blood cell. Okay. And then the other question there, which is kind of probably one of the more important ones, why does the bicarbonate need to leave the cell in the first place? Like why even create this whole chloride shift thing? Why not just let the bicarbonate stay there, right? There's no reason to create this whole system. Well, there is a reason. The reason is that for anytime we have a reaction like this, we have to get rid of the products if we want that reaction to continue, right? So if we just let the bicarbonate build up at the end there in the red blood cell, then the reaction would stop, right? Or it actually would start going back the other direction, right? This is the same thing with the electron transport chain that we talked about when we're making ATP and those electrons are going down the chain, and then they have to bind to oxygen, and then oxygen can take them away in order for that to continue, right? If we just let the electrons build up at the end of that chain, then we don't have a gradient anymore for electrons. That reaction is going to stop, and we stop, right? If we don't have oxygen to do that. So it's the same idea with this. If we let the bicarbonate build up at the end, then this reaction stops, and we don't let any more CO2 into the cell, and we can't transport CO2, which is the whole purpose of this thing, right? So we need to, we'll just say, to keep the reaction going, we need to remove the product, or the products, okay? So we need to get rid of those products of that reaction in some way in order to keep this reaction moving. Okay, any questions on that first one there? Okay, all right, so that's the first one. Second one's just going to be mostly the opposite, okay? So if the other one was the tissues, obviously this one is the lungs. All right, where does carbonic anhydrase work? The same place, okay? It can drive the reaction the other way too, right there, wherever we see this, that reaction happening in the red blood cell. Okay, what happens with the hydrogen ion now? So right, the hydrogen ion was bound to hemoglobin, but what do we see here? We see oxygen coming into the cell, and here's the hydrogen with the hemoglobin, and there's so much oxygen that the oxygen kicks that hemoglobin, or kicks the hydrogen off of the hemoglobin, and now we get oxyhemoglobin instead of the hemoglobin with the hydrogen bound to it. So that kicks the hydrogen off, and it can go right back into that equation now. We need it again to make CO2. If we don't have the hydrogen, we can't make CO2 again because we can't do this equation, right? So that hydrogen goes straight up. It's going to go through that reaction again, okay? So I'm not sure how to write that in a short way under what happens to hydrogen, but I guess I should try. So we'll say it gets kicked off of hemoglobin by oxygen, okay? And then goes into the bicarb reaction. Okay, so then it's going to go up and go into that bicarbonate reaction again, okay? So now we see our chloride leaving. That's that same chloride that came in down at the tissues. It can leave now, but it does that in exchange for bicarbonates coming back into the cell. Okay, so comes in, then binds to hemoglobin, or not to hemoglobin, but to hydrogen, okay? And then that turns into carbonic acid. That can turn into CO2 and water. And that's how we get our CO2 that we can breathe out of our lungs. And the chloride just goes out of the cell. Okay, so again, this is kind of, this equation helps us both with buffering of hydrogen ions, but it's also incredibly important for transportation of CO2 throughout the body because of this. Okay. All right, any questions on that? So that's the chloride shift. So you'll definitely want to understand kind of all these questions around this, be able to understand what's going on in those different parts of these two diagrams here at the different locations. Okay. All right, so let's go ahead and do this question here. All right, so take your pick there. Give you about 15 more seconds. All right, anybody else? Give about 5 seconds. Go ahead and send it in if you haven't. Okay. All right, see what you guys said. Okay, so most of you are saying B. Okay, a couple saying A. Let's take a look there. So at the tissues, okay, at the tissues, bicarbonate goes out of the red blood cell and CO comes in. Okay, let's make sure that's right. So at the tissues here, we see bicarbonate going out and chloride coming in. Okay, so that's correct. And that bicarbonate came from excess blank and hydrogen and water combining. So that should be CO2. Okay, so all that CO2 was coming out of the cells, combined with water, eventually turned into bicarb and hydrogen ion. Okay. All right. Any questions about any of that with the chloride shift and the transportation of CO2 before we kind of transition over to ventilation control for the next half of class? Okay. All right. So a couple different locations in the brain where we control our breathing. Number one, the medulla oblongata. Okay. There's what's known as the ventral respiratory group. Okay, which is kind of right next to where the pons and the medulla junction. And there's a couple important nerves that are kind of controlled by this part of the brain. So the phrenic nerve, which is the nerve that innervates the diaphragm. Okay, and then the intercostal nerves, which are important for contracting those external intercostal muscles that cause our rib cage to lift up, right, during inspiration. Okay. So the function of this part of the medulla, this what's known as the VRG, the ventral respiratory group, is it's really what drives inspiration by activating those muscles, right? By activating the diaphragm and activating the external intercostal muscles. Okay. Also gives some rhythm to breathing, and then also drives expiration just by causing those muscles to relax, right? Because that's what we said. Resting expiration is just simply those muscles relaxing. Okay, and then we have the dorsal respiratory group, also in the medulla oblongata, important for the chemoreceptor reflexes that cause us to breathe more, right? So when we have more CO2 in our blood, that causes us to breathe more, and that's kind of controlled by this part of the brain. And then the pons is involved by smoothing out our breathing for us. Okay, and so you can kind of see those regions here. So the ventral respiratory group, dorsal respiratory group, all part of the medulla there. Okay, and then you can see how they come down and interact with the phrenic nerve for the diaphragm, and then for those external intercostal muscles there. Okay, so let's talk about those chemoreceptor reflexes a little bit. And so there's kind of multiple locations where chemicals are sensed throughout the body. Okay. So there's central receptors that we find in the medulla oblongata itself, and those are actually sensing the amount of certain chemicals in the cerebral spinal fluid, the CSF. Okay. And then there's peripheral receptors as well, that we find those in those same two locations that we found the ones that are responsible for controlling blood pressure, right? In the carotid and aortic bodies. Right, so right there where the carotids split into internal and external, and then right there where the aorta comes up and turns. Okay, so we're sensing the level of these chemicals in the blood in those two locations. Okay, so the chemical that's the most important, the one that these are sensing, is CO2. Okay, so CO2 is known as the primary driver of our ventilation. So these little receptors are there in the medulla sensing the level of CO2 in our cerebral spinal fluid, and then they're there in the aorta and the carotid arteries sensing the level of CO2 in our blood. Okay, they also detect hydrogen and oxygen, but CO2 seems to be the main driver of this. Okay, and so these things are very similar to the same things that cause vasodilation when we talked about blood pressure control, as well as the same things that cause that rightward shift of the curve, right? So things that would be associated with doing more metabolism would also cause you to breathe more, right? That makes sense. Those same things also cause your blood vessels to get bigger, and they also cause that rightward shift of the curve, which would cause more oxygen to fall off. all of that just makes sense, right? You don't really have to memorize that if you kind of just think about it, and it makes sense, right? So more CO2, more hydrogen, and less oxygen, all those things are going to cause us to breathe more, right? But if we go the opposite way with those, less CO2, less hydrogen, or more oxygen, then we need to breathe less because don't have a buildup of byproducts of metabolism, and we've got excess oxygen around. Okay, at least on the CO2 and hydrogen side of those things also cause, so high CO2, high hydrogen, those both cause vasodilation as well, and they both cause a rightward shift of the curve. Okay, all of those things make sense because that means we need to deliver more oxygen if we're doing more metabolism. Okay, so that's part of what controls our breathing. Okay, go ahead, right?
So how some people can hold their breath way longer than other people? Does that have anything to do with the chemoreceptors and just make you want to breathe?
Yeah, it's a good question. Brett asked, why can some people hold their breath longer than others, and does that have anything to do with these chemoreceptors? And yeah, my guess would be that these are all reflexes, right? And reflexes can they happen without the involvement of the brain. That's the idea of a reflex. It just goes to the spinal cord and back. But in this case, or with any reflex, the brain can modulate the reflex, right? Like when you guys are doing those patellar tendon reflexes, and you didn't have anything else to do, and you just kind of looked at it, you didn't have as big of a reflex as when you were counting backwards, right? And so it's kind of this idea that when your brain's distracted, you have stronger reflexes. So it could be kind of something related to that, where you kind of train your brain in a way to modulate those reflexes. So that's part of it. Other parts of it have to do with just training your body not to move, so you're not using the oxygen, right? Training your body to relax, so your muscles aren't tense, so you're not using the oxygen. So there's a lot of kind of just behavioral training that people do that really know how to hold their breath for a long time. Are the chemoreceptors different? I'm not really sure about that. Yeah, but I know there's some of those other things. Sure. Yeah. Good question. Other questions about any of that? Okay, so a few kind of applications here with chemoreceptors. So when you see somebody who is breathing 100% oxygen, right, or somebody has an oxygen bottle attached to them in some way, and there's multiple ways to deliver that oxygen. So one could be a nasal cannula, like this one. Okay, you could also have a bag valve mask or a non-rebreather mask. There's different versions of this. See, this has oxygen attached to it like this. Okay, in both of these cases, even though it might be 100% oxygen in that hose, the person isn't breathing in 100% oxygen, right? Because with this case, obviously it's not a perfect seal in her nose, right? And she can also open her mouth and breathe too, which she most likely will. So she's not getting 100% oxygen. In this case, in the case down here with this type of setup, you actually breathe out some air into that bag, and so you're actually breathing back in some of your own CO2 and some of your own air. And so the oxygen that's coming in helps to give you a higher oxygen percentage, but you're still not getting 100% oxygen. Okay. Yeah, and even in that case with this non-rebreather mask in emergency situations, you might still only be getting about 60 to 80% oxygen. Okay. And the reason for that is breathing in 100% oxygen is dangerous, because what happens is it completely kind of dilutes out the CO2 in your lungs. Okay, which can make your CO2 levels drop in your blood. And if you don't have enough CO2 in your blood, and you go back to these chemoreceptors, if you don't have any CO2 there stimulating your chemoreceptors for you to breathe, you just stop breathing. And that's really what happens with hyperventilation, right? So when somebody breathes so much over and over and over again, when they don't really need to breathe that much, then they're just blowing off all of their CO2. Their CO2 levels are dropping. And then they lose that drive to breathe because you don't have that chemoreceptor reflex working. Okay, so giving somebody 100% oxygen and just sealing off that oxygen and letting it all come in can actually make them stop breathing completely. And so that's why there's just these different methods that don't actually give them that much oxygen. Okay, so yeah, same thing with hyperventilation. You're lowering your CO2 levels so much that you lose that drive to breathe. Okay. And what do you do when somebody hyperventilates? What's the treatment for it? What do you guys seen it in movies, learned it in a CPR class? What do you do? Yeah, do a paper bag, right? They breathe into a paper bag. You're literally trying to get them to breathe their own CO2 back in. That's the whole point of that, right? Is they're breathing out CO2, but you don't want them to lose that CO2 because it's already too low. So you have them breathe that back in, so their CO2 levels start going back up. Okay, and a paper bag is important because it's not totally sealed, right? So you're not suffocating them, right? But paper bag kind of allows some air to move, but you want that CO2 to come back in so you can get their drive to breathe going again. Okay. All right. Any questions on that? Okay. All right. So a couple terms, definitions here. Hypernia versus hyperventilation. Hypernia is increased ventilation in response to an increase in O2 demand. Okay. So this would be like in response to increases in metabolism. So when you start exercising, for example, your metabolism increases and you need to breathe more. Right. So that's just the kind of a normal increase in breathing. But then we can also have hyperventilation where we start breathing more, but we actually don't need to. Right, there's actually no increase in O2 demand, and so this can be either because of voluntary breathing or some kind of psychological stress, where we panic attack, anxiety type things like that cause you to breathe more, but you don't have extra metabolism, so you're not breathing out CO2, like extra CO2 that was being made. You're just breathing out the CO2 that you had, and that's why your CO2 levels can drop so rapidly. Okay. All right. So with exercise is definitely a form of hypernia, not hyperventilation. Okay. We can see it somewhere around a 10 to 20-fold increase in ventilation with exercise. Okay. And the main things that drive this, well, number one is those CO2 levels. Okay. And there's chemoreceptors out around your muscles as well, in the arteries around your muscles. And so that would drive it. There's also psychological stimuli. Even before you actually start exercising, your brain is preparing for that exercise by actually causing you to breathe a little bit more, and your heart rate goes up a little more when you're tying your shoes, when you're getting ready. It's all called the anticipatory response. So there's definitely fully psychological stimuli outside of those chemical factors. And then the interesting part about this is it's pretty well known that proprioceptors, so muscle spindles, Golgi tendon organs, those things that are going to be sensing more tension and more stretch when you're moving, those are somehow related to the breathing response as well. We know this because certain spinal cord patients that exercise don't have the same increase in ventilation that people that have full access to their to the sensory information come in that way. So it's kind of an interesting thing. We don't have a good grasp on how all of that works. We just know that it's related. Okay, so a little bit about altitude, high altitude. Okay. So this is a picture. Anybody know what picture this is actually, or what mountain this is? Look familiar to anybody? Anybody on the chat? We'll put the name of that mountain in. Got any mountain climbers in class? Maybe not. It's the highest peak in the 48 contiguous United States. This is Mount Whitney right here. Peak right there. Mount Whitney. It's in the High Sierras. It's only about a 6-hour drive from here to hike Mount Whitney. You have to get on the lottery list to get a permit, like about a year in advance. But yeah, it's the tallest mountain in the 48 contiguous United States. There's a Mount McKinley in Alaska is higher than this. That's why there's higher in the non-48, in the lower 48. But it's a great hike. It's about a 22-mile round trip hike, but it gets up to about 14,700 feet. And so if you're interested in anything like that, it's definitely a fun one to train for and go for it. But yeah, so what happens when we go to a high altitude like this? Okay, first of all, we get a big increase in our ventilation. Okay, why does that happen? Well, it happens because our oxygen levels in our blood drop a little bit. We get lower arterial oxygen concentrations. Okay, so that stimulates breathing, right? Because that's one of those things the chemoreceptors are looking for, is low oxygen. Okay, so but without increasing our metabolism and we're breathing more, what's going to happen to our CO2? It's going to go down, right? This is what we were just talking about. Okay, CO2 goes down because we're breathing extra. And then what would that do to our pH? That would make our pH go up. That would make our hydrogen ion concentration go down. Because if we're losing CO2, that's directly tied with hydrogen ions because of that equation we were just looking at. Okay, So what would happen if our hydrogen ions go down, our CO2 goes down? Well, what effect would that have on that oxygen hemoglobin dissociation curve? Well, it does the opposite of what we were talking about before. Before we were talking about the rightward shift. This would be the opposite. This would be a leftward shift now. Okay, so we get easier loading of oxygen at the lungs. This makes it a little bit harder to unload. Okay, but this is actually helpful, right? Because if your blood is a little bit desaturated, it doesn't have as much oxygen in it. If we shift that curve to the left, then it gets a little bit more saturated, right? And so this is a helpful thing that happens here. Okay, for example, your blood at 19,000 feet elevation, if you put that little thing on your finger with the red lights, it would only be about 67%. Okay, that's a lot lower. Now, if you measured somebody's O2 sats at sea level and it was 67, then they'd be in trouble, right? In major trouble, major respiratory distress, most likely going downhill quickly, right? But this would just be a perfectly healthy person just sitting at 19,000 feet, right? But when we shift that curve to the left a little bit, that saturation would go up for them. So, okay. One of the effects of this, that low oxygen, is it stimulates our erythropoietin production. That's that. Oh, sorry, I forgot the left was there. That stimulates our erythropoietin production. Remember, that's that hormone that gets released by the kidneys that then causes our bone marrow to start making more red blood cells for us. And that increases our hematocrit. That doesn't happen automatically. That takes 2 to 3 months to actually see that change. So it's not like that would change automatically. We increase those red blood cells slowly over time. Okay. All right. Okay, so a couple diseases related to this. COPD is kind of a large chunk of diseases that really involves 2 underlying diseases. It can either be emphysema, or it can be chronic bronchitis, or it can be both. When a patient's diagnosed with COPD, that's what it means. It means either emphysema or chronic bronchitis, or they got both. Okay, so risk factors you might imagine: smoking, genetics, and then there's those common factors there. But let's look at each one. So emphysema, we've kind of already talked about. It's the breakdown of these alveolar walls. Okay, and so we have a real hard time exhaling because we can't smash down these alveoli. Okay, and so that's kind of one we already talked about with emphysema. Okay, chronic bronchitis we haven't talked about, but the idea with this one is there's long-term exposure to some kind of irritant in the lungs, which produces lots of mucus, and then that mucus just stays there. Okay, excessive mucus in the airway. And you can kind of see what would happen if there's this big ball of mucus in one of our smaller tubes, then that eventually that can completely block off flow into one of our alveoli. That air gets trapped in there. Okay, so that's pretty much a part of the lungs that you can't use anymore when you get a little part of it that's blocked off. But more dangerously, that also leads to many infections, and these people can get pneumonia pretty quickly. Okay, so that's chronic bronchitis. And this just kind of shows you a breakdown of those 2 diseases leading to COPD and kind of what they typically look like. Okay, lastly, we'll just finish up with this. TB, or tuberculosis. Most of you have all probably had a TB test before, right? Where they put that little needle under your skin and put a little bit of irritant in there. And if your body reacts to it, then they know that you have TB in your system. Right, and so what it is it's a bacteria, okay, and it forms these little nodules in the lungs. Okay, it's estimated that about 1/3 of the world's population has TB, but most people never know it. It never becomes activated in most people. Okay, so it can kind of sit in this inactivated state for a long time, and then all of a sudden kind of become activated. Okay, and usually sometime when the immune system is compromised for some reason. Okay, so it really just gets in and just destroys alveoli completely, causes spitting up of blood, night sweats, and eventually leads to death. Antibiotics can definitely treat these, but there are certain forms of TB that are antibiotic resistant now that are very hard to treat. So, all right, guys, that's all we got for that. That's the end of the material for exam 4. So we'll see you guys on Wednesday. And have a good rest of your day.